Okay, yeah, so thanks, Camilla. Yeah, so I worked here over summer for 2 months, where we were just like getting started on looking at focused ultrasound. I was mostly sort of looking at simulating the ultrasound field in order to target certain areas. So I've actually carried on doing that at the University of Nottingham, where I've been for about 3 months working on these computational models. So I'll give a bit of an introduction into the technique, and then I'll talk about sort of my work into the simulations as well. So first, we just want to maybe introduce like ultrasound, the physics behind the technique. So ultrasound is part of acoustics. It's pretty much the same thing as sound. It's waves traveling through the air, carrying energy in the same direction as the oscillations. And ultrasound is a higher frequency than humans can hear, hence why it's called ultrasound. And the sort of frequencies we use with this stimulation is anything above 100 kilohertz. So the most common use of ultrasound at the moment that everyone will sort of be familiar with is ultrasound imaging, where a transducer emits an ultrasound wave in bursts into some tissue, and based on the reflection, absorption, and transmission of this ultrasound, we can use it to create images. Now, these transducers, this is what emits the ultrasound waves, and they use the piezoelectric effect, which is basically, there are certain types of crystals that when you apply a stress, in other words, when you compress them, a voltage is induced over each side. But this works in the other direction too, and we can apply an alternating voltage in order to cause an oscillation in the size of this crystal. And what that does is causes oscillating pressure, which emits these ultrasound waves in the medium. So we can use that, and usually these are constructed in the sort of shape of a plate or a bowl. So you can see on the left image here, that's the pressure distribution due to a planar sort of plate transducer. But what we're interested in with focused ultrasound is these curved transducers that really concentrate the signal into a very sort of narrow region. So they sort of take this characteristic elliptical shape. And this is sort of on the resolution of millimeters. So yeah, now we've given a little bit of a background on the physics of ultrasound. Why do we want to be using this for neuromodulation? Well, sort of, first of all, it's non-invasive. The levels we're going to, operating at for neuromodulation, it's also safe, or at least no one's found any unsafe results yet. It's really precise. So compared to TMS, for example, which, it's hard to concentrate on a very specific brain area, due to the millimeter precision of ultrasound, we can really get specific. And also, due to the radius of curvature of some of these bowl-shaped transducers, we can get quite deep, potentially up to about 10 centimeters, which is, I think, plenty to target sort of quite deep structures. And we can also give personalized treatment, because these acoustic simulations that I'll talk about later take an input of individual MRIs. So we can basically get any patient in, take a scan, and see where we're going to be targeting from that. Now, there are a couple of limitations. Probably the most obvious limitation is the fact that the skull is quite a big absorber of ultrasound. So due to the cortical structure inside the skull, a lot of the ultrasound sort of scatters and is absorbed and is reflected and deflected, so that we really have to work on these simulations to ensure that we're getting the right amount of pressure in the right location. So this is an example of a simulation showing the targeting of the thalamus. And you can sort of see the focal effect and also the sort of deep penetration into the brain. So yeah, it's quite, in terms of that, it's a pretty good technique. So what's been done so far? Well, there's been quite a lot of animal studies, and it's sort of in the last probably 5 years or so, human studies have started to happen. So at the moment, they've been sort of targeting the motor cortex, visual cortex, and actually they've found that with the primary visual cortex, phosphenes have been recorded. Thalamus, caudate, and somatosensory cortex, where tactile sensations have been felt in like individual fingers. Now, no like more significant physical responses have been seen in humans, but in rats and mice, I think there have been twitches of limbs and tails due to targeting the motor cortex. And I'm not going to go into like loads of detail into these studies, but I'd recommend the following two review papers if you're interested. I think there's also another one coming out this year that will give a really good rundown of all the sort of previous research done. But everything's sort of, a lot of the human studies have really just been these last 5 years or so. So it's really exciting field. Okay, so I've talked about the physics and we've talked about the responses, but how does the ultrasound actually modulate neural activity? Well, this is actually a really active area of research and we're not 100% sure yet what the mechanisms are, but I'll give a sort of rundown of a couple of potential mechanisms. So this is just a load of suggested mechanisms, but I think personally, I think the middle three are the most important. So we've got the fact that ultrasound can open mechanosensitive ion channels. We've got thermal modulation as a result of increasing temperature. And there's also cavitation, which is the creation of small microbubbles within the membrane that cause the membrane to sort of stretch and the potentials to change. So these are probably the most common proposed ones. However, thermal modulation, with the sort of setups that most of these experiments are running, and actually within the safety limitations, we don't tend to raise the temperature high enough for thermal modulation to be important. And actually, in a study on some C. elegans neural cells, when wiping out some thermosensitive ion channels, there was no change in neural response, but the mechanosensitive ion channels did create a response. So we think we're going to be concerned with mechanical effects. In other words, the tissue wiggling around a little bit. So what can we tell about these mechanosensitive ion channels? Well, there's been a really recent study that actually just came out this year where it's been found that it's calcium-gated sodium channels that are responsible for some of the neural response. And first, sort of wiping out these sodium channels, they've shown that it is in fact responsible. And in this paper, Yu et al., they went further and genetically wiped out individual mechanoreceptors in order to figure out like which ones the ultrasound is interfering with. And yeah, as you can see here, the sort of TRPP1 and 2 and TRCP1 were responsible in this study. Now, again, it's still kind of like early days with working out the mechanisms, and this also doesn't rule out any other mechanisms happening at the same time. So cavitation, like I mentioned earlier, these bubbles forming is still quite a promising thing to look at too. And there's a lot of like computational models that have shown sort of matching with cavitation too. Okay, so that was the mechanisms. And then finally, sort of before we go into the simulations, when we're running these experiments, a bit like TMS, we're going to be sending in bursts. And I think this is originally modeled after sort of the TMS protocols. But in terms of how we send in the pulses, I think personally, I think there needs to be a bit more research into what parameters we want to use, which ones are most effective at eliciting a response. And also, it's worth saying that TMS is a more direct technique of stimulating the electrical activity, whereas ultrasound requires all these other mechanisms that could potentially cause the stimulation. So I think a lot more needs to be done into figuring out exactly the optimal sort of protocols that we're going to be using. But for now, I'll get on to talking about some of the simulations. So why do we want to run simulations? Well, first thing is safety. So I mentioned all of these mechanisms. These mechanisms can also be responsible for tissue damage. So if you put too much pressure into the tissue, you can cause these microcavitations to increase so much that the bubbles burst, or you can just cause rupture of the cells. And if the temperature is too high, well, the thermal effects also could cause tissue damage. So we want to make sure by running acoustic and thermal simulations that the pressure is not too high and that the temperature is not too high either. But one of the most important things about simulations is being able to determine that we're actually targeting the right part in the brain. And then we can do this based on individual anatomy. So what tools can we use? Well, I've been using K-Wave, which is a way of solving acoustic equations specifically for medical ultrasound. It's just a MATLAB toolbox. It's good, but what's coming out this year, hopefully, is a sort of more integrated package where anyone who, whether you know about acoustics or not, or know how to use MATLAB, can plan ultrasound therapy. And there's other tools as well, but for what I found, this is sort of it runs in the quickest time, and it's accurate enough. So so this is sort of like a typical workflow that I've been working on. So you take your MRI images or your CT images. You can either put in the whole head or just the skull, and a source field, which is essentially what comes out of the transducer. So you can plug those into the simulation, and then you see at the top we get the acoustic simulation. You can really see the sort of focusing. But I also just want to draw attention to the reflections that happen just now when it comes back off the other side of the skull. One reason why we're trying to do these models is because the reflections are actually quite important as well. We might end up with other hotspots or other standing waves as a result of that, and we want to make sure that we're not trying to stimulate the wrong part of the brain, essentially. And the thermal simulation, it doesn't look like much, and that's because we don't really heat up the brain that much. It's the skull that heats up the most, just because it absorbs so much ultrasound. So once we've run those simulations, we check that we've hit the target, we check safe levels. And what we're particularly interested in doing at Nottingham is we then want to find a way of inputting these results into neural models and trying to predict whole brain effects of targeting a certain region. So this is sort of what a source input might look like if you put it straight in the software. But I was having a lot of problems over summer in trying to actually accurately represent the transducer in the software. So, and that's because, this is what a sort of normal transducer this is the sort of normal pattern that it gives. But our transducer here at the CBU and also at Nottingham is made up of these rings. And what's the significance of that? Well, if we have these rings, we can input a different signal into each ring. And what that means is we can steer the array using different phasers to either go towards the focal plane or away from the focal plane, which means that we're actually shortening or lengthening the focal length. And what that means is we can target deeper structures or structures nearer the outer edge of the brain. And this is sort of a result of changing the phases. We can get this like really short focal length, but then we also can get really long ones. And actually this sort of shows both. This is just a model. We can calibrate it to make it a lot more effective. So this was sort of my the problem I was stuck on a lot of the summer, is trying to simulate this. But we've kind of settled for the fact that we can't simulate an ultrasound transducer too well. So what we're going to do is construct a tank and a scanner so that we can measure the exact pressure field coming out of a transducer and plug that straight into the software. So what we can also do then is verify that these models are accurate and that the predictions that we have are actually what happens in real life, as well as whether the targeting is exactly how we predict too. So we're going to use a phantom skull for that. So I just wanted to also give a little bit of a limitation of using this sort of simulation, in that when we're using the input into these simulations will probably be MRI images for healthy subjects, because I'm not sure you can really CT scan, healthy subjects too easily. So what we get from an MRI scan is essentially a segmented binary map of the skull, and it looks a bit like this sort of bottom image where it's just going to be a single sort of tissue type, and all we can do is assign sort of average skull density values, skull speed of sound and everything. We can't really describe the sort of detail that the actual bone has. So there are limitations, and we just have to be aware that we're not always going to be giving the exact values, but I think when it comes to simulations, we always look for the worst case scenario, just so we're not going above safety limits and everything. And hopefully when we get to verify these models in a tank, we should be able to sort of fine tune it so that we're actually using the right parameters. And another limitation is shear waves. So I didn't mention them earlier. Shear waves are like side-to-side waves that only exist in solids. So when the ultrasound goes through the skull, it actually splits off due to the difference in refraction angle, and it gives us slightly offset waves. And what that means is that our K-wave model only uses longitudinal waves, not shear waves. So we have to just be aware that there are some slightly differing effects due to these shear waves. But in the focal region, we're still going to be predicting the right focus. We just need to be aware of it for all the behavior that goes on away from that. And on the right is actually a study that showed how these shear waves propagate around the skull and potentially into the ear channel, causing an auditory confound. So when running these experiments, we actually have to play a sound over some headphones that masks that sound that comes from the ultrasound sort of traveling through the skull. Okay, so that's sort of a very quick background on the simulations and the mechanisms. So at the moment, I'm working on a project led by Marcus Kaiser in Nottingham, where we're essentially looking into using focused ultrasound to intervene with brain and mental health. Specifically, we're looking at epilepsy, I think, in the long term. So the first thing that we're going to do is model this ultrasound propagation and also work on some brain models, some network models, and then we're going to try and figure out the interface between the two, and then validate these ultrasound models in a tank. And then we've got some non-human primate data that we've got from Newcastle that we're going to use to validate some of these predictions. And then we're going to do a pilot study in humans targeting the sort of main areas that I talked about earlier that have been done before. So we essentially just want to check that our equipment works and that we can elicit a response in the way that other people have. And we're going to do, pre-test structural MRI for the simulations, and then fMRI and diffusion weight imaging, and then fMRI whilst in the scanner to monitor online effects. Now, while I mentioned online effects, one of the really interesting things that we want to work on in the long term is the offline effects of focused ultrasound. There have been probably 1 or 2 studies looking into the offline effects, so not much at all. But it has been shown in one study to increase neuroplasticity and functional connectivity for up to a month or 2 after the application of the ultrasound. So not really much known about it at the moment, but really exciting to think about, especially when we're talking about epilepsy, for example, changing the functional connectivity. So yeah, very brief rundown of what we've sort of been working on. To conclude, focused ultrasound is a really precise alternative to other NIBS techniques. But we don't fully understand the mechanisms. And while we've observed effects in animals and humans, a lot more needs to be done, especially in other brain areas too. We need to run these simulations to ensure safety and correct targeting. But yeah, like I say, a lot more research needs to be done, especially into burst parameters, offline effects, and other brain structures. So I'd just like to say thanks to AJ and thanks to Rick for supervising me over summer, and also from the maths department, and then my current team in Nottingham as well. So yeah, thank you very much.